3-Minute Thesis, Cornell University, 2018. Divya Shiroor, Regeneration 101, Lessons from Flatworms.
If I told you you could learn to play the piano by studying under Cristiano Ronaldo, Beethoven, or Madame Curie, who would you pick? Most of us, if not all of us, would definitely pick Beethoven. Why? Because he was so good at it, and there's a certain merit that comes from learning from the very best. This is the exact reason why I study regeneration in flatworms called planarians. Planarians are known as master regenerators. On the screen here, you have a worm that has been cut into 3 pieces. Within 14 days, every individual piece has regenerated into a fully functional worm. Now, regeneration in these worms is under the control of special cells called stem cells. Stem cells give rise to other cells that then make up organs and tissues in the body. Now, our organs and tissues have stem cells as well, and we also share many of the same genes as these animals. Why is it then that these flatworms can regenerate so fantastically in ways that no mammal can? This is what I hope to understand during my PhD. For my dissertation, I am studying how stem cells in these flatworms respond to injury. To do this, I amputate or remove different parts of the animal and observe the stem cell response. What I found so far is that number 1, as early as 48 hours after injury, the stem cells congregate around the wound site. Number 2, this is not unique to one particular injury. Many different injuries are capable of invoking this response. What makes these results really cool, though, is that I found that not only healthy stem cells, but also stem cells that have been subject to severe pressure respond just as efficiently in this manner. Now, normally when a stem cell has been subjected to severe pressure, it dies. However, I have found that in the face of injury, this severely stressed out stem cell seems to be receiving a signal that tells it, hang on, you can't die. You need to survive and do your job as a stem cell and go regenerate that missing tissue. I am now working on uncovering what this signal might be that drives these stem cells to respond so efficiently to injury. Given that we share so many of the same genes as these animals, I hope that by uncovering this signal, I might be able to uncover pathways that we could someday use to stimulate stem cells in our organs and tissues to respond to injury just as efficiently as in these animals and perhaps bring about regeneration. By studying regeneration in an organism that is so inherently good at it, I hope to learn from the very best. Thank you.